Hello, welcome to this module on DNA replication, cell cycle and mechanisms of cell cycles. Now, I'm Dr. Subradip Karmakar. I'm an assistant professor in the Department of Biochemistry at All India Institute of Medical Sciences, New Delhi. Now, today we're going to touch a very important aspect of cell and molecular biology, that is DNA replications and the mechanism of the DNA replications. So before we proceed into going in detail about the DNA replications, we just need to brush out a few basics about it, because that is going to be extremely helpful for us to understand the subsequent slides of this particular module. The first is that DNA is a genetic material. The second is the structure of the DNA. The third is the details of the DNA replications. The fourth is replication enzymes, that is the enzymes which assist in the process of the replications. And the last but not the least are the prokaryotic replications and the eukaryotic replications. Remember that the DNA replication mechanism is different if it is a prokaryotic bacterial system as compared to an eukaryotic mammalian system. Coming back to sets of experiments which are actually been done. So this DNA replication is actually a science that was enriched by people from different avenues and trying to do some basic but extremely crucial experiments and then trying to enrich our knowledge about this particular area. Now, first is that how to know even that the DNA is a genetic material. So people has a confusion that proteins as a genetic material and RNA is a genetic material, which is true, right? RNA can act as a genetic material, but how to know that DNA is also a genetic material? So in order to do that one, people did a very simple experiments. Is that they took a bacteriophage and they took the bacteriophage and allowed that phage to infect a bacteria. And what they did is that they took this bacteriophage and labeled the DNA of the bacteriophage with a particular nucleotides, and then the proteins of the bacteriophage with a particular radioactive nucleotides. Mostly the convention is that we label with phosphorus 32, P32, which is a gamma emitter for the nucleic acids, and sulfur 35 for the proteins, because methionine in the proteins contains sulfur. So label them with sulfur, radioactive sulfur is 35, and DNA contains nucleotides and then the phosphodiester linkages and phosphate backbones. So label that with the P32, which is in beta emitters. Now they did both of these two. They label the proteins with S35 and they label the DNA with P32, and then they allowed this phage to infect into the bacteria. And when they allowed the phage to infect into the bacteria, then they found that the genetic material which has been found within the bacteria is all P32 level DNA, but there is no S35 level proteins. And what does that mean? That means that the material which has been transferred actively from the phage to the bacteria is actually the nucleic acids, which by then that these nucleic acids are the one which is the units of hereditary, because when the proteins are actually been there, then there is no replication. The bacteriophage is been infecting the bacteria, but there is no further replication of the bacterial phage. But the moment when it transfers this nucleic acid, then there is a replication and then there is an increase in the number. And that clearly points out that the material for genetic inheritance or the genetic transmission of information is actually the DNA and not the proteins. So with this very elegant experiments and radio level nucleotide leveling experiments, people could actually say that DNA is the genetic material. So the DNA replication process. Now once we know that the DNA is the genetic material, the next question is that how does the DNA actually copies it? So you know that when there is a parental cell and then there is a daughter cells, now the DNA is actually transferred. Now what happens if the DNA is actually transferred from the daughter cells to its own daughter cells? Okay, again the DNA will be transferred, but just try to imagine what happens in a bacteria which grows extremely fast. If the DNA actually goes on, keep on shedding every time the daughter cells is actually produced or birds of, then a time will come very quickly that there will be no more DNA in a particular cell, because it will be completely diluted out, right? So that's not going to happen. Now that really doesn't happen in the real life scenario. So there is a mechanism by which the DNA is actually being copied and multiple copies are actually being produced, and whenever the progeny is actually being produced or the daughter cells are actually being produced, then a copy of the DNA which has been produced goes to that particular daughter cells, making sure that never the DNA gets diluted, never the DNA runs out of its copy number once. It doesn't matter how many number of cell divisions it undergoes. And therefore the main problem is to that how to understand the mechanisms of the DNA duplications and the DNA replications, right? So people do some series of very interesting experiments, and before that, actually just try to tell you for a few minutes that there are actually there is a theory that actually explains that there are three different mechanisms by the DNA could replicate. One is known as the conservative replications. The concept of that is that we have the two strands of the DNA, and two new strands actually been generated. So the parental strands and the daughter strands are completely, one is the old sets, another is the new sets. So that's the conservative replication mode, where totally two new strands of the DNA are actually been synthesized from two old strands. Now there is a semi-conservative mode of replication which tells about that, look, there is two strands of the DNA, but one of the strands is going to act as a template and is going to create a new strands, and then it prompts or produces a hybrid strand, and so forth. That's known as the semi-conservative mode of the replications. And then there is a dispersive mode or a random mode or a mixed mode, whatever you can call of, where it is been said that it is both semi-conservative as well as it is also conservative depending upon the different regions of the DNA. So you have three main modes: the conservative, completely new strands are produced; the semi-conservative, where hybrid strands are produced; and a dispersive mode or a random mode, where both of them are actually produced. People do some very elegant experiments to prove that the DNA replication is actually semi-conservative mode. That what is actually the semi-conservative mode of the DNA replications. Now this is the Michelson-Stahls experiments, and they did an elegant but very simple tools and techniques, and these are cesium chloride based density gradient centrifugations. Okay, and in these centrifugations they could very nicely piece together and say that okay, the DNA replication is semi-conservative mode. What they did is that they leveled the DNA with two different kinds of nucleotides, the nitrogen 14 and nitrogen 15, which is actually the two different radioactive nucleotides of the nitrogen. And then they actually allowed the cells to replicate, and then eventually if it's a conservative mode of replication, then they should see, there's because there's two nucleotides has actually defined molecular masses, they're going to settle differently in the cesium chloride density gradient, and you could see actually at the end of the experiments that there is a hybrid N14 and N15 bands that are produced. And this hybrid bands clearly points out to the fact that there is a semi-conservative mode of the replication which is taking place, because one of the strands is being exchanged and the new strands is being produced, and the old and the new strands are being forms and hybrids and together. And that is the reason why you see both an N14 as well as an N15 signature in that cesium chloride band, and that actually proves beyond a reasonable doubt that through these Michelson-Stahls experiments that there is a semi-conservative mode of the DNA replications, and now it has been well accepted in the community, and we don't have any dispute in that the DNA do replicate in a semi-conservative. Now after that we are going to talk about a little bit on the structure of the DNA. Now after Watson and Crick has actually discovered in the elegant experiments in the elegant way that the DNA is a double-stranded helix, then this information of the DNA itself is very important, because the moment we say that the DNA is double helical structures and that one strand of the DNA is complementary to the other strands of the DNA, this immediately implies us that one strand of the DNA can act as a template and can actually produce or synthesize the other strands of the DNA. And therefore the essence of heredity actually erupts or emerges from here, that if you just have one strand of the DNA, that has sufficient information to dictate the synthesis of the other strands of the DNA. And that is fantastic development and fantastic interpretation out of just the structural solving of the DNA, that how the DNA can act as a template and produce daughter strands, and all the information to produce the daughter strand is being contained in just one strand of the DNA. Now some of the facts about the DNA replication is that why do we need to have the DNA replications? Now as I mentioned just a few minutes back, that when the cell divides, if the DNA doesn't replicate, doesn't copy itself, then it's going to soon dilute out, right? And then once it dilutes out, you will have a cells without any DNA, and there's not going to be a viable cells. So we need the viability of the cells, and there's an efficient way to do that is to replicate also the DNA. In addition to replicate or produce or double the other components of the cells, then the cytoplasmic content, which is also very important. The another very important thing of the DNA replication is that most of the time, there is different kinds of assaults on the DNA, like the DNA strand breaks and DNA replication that takes place, and during the time there is a strand breaks, and then there is a DNA repair system. So all of these processes actually has to be compensated by a mechanism to replenish the DNA which is been lost, or which has been altered. So DNA replication is a very efficient way to do that particular alteration, so that the genetic material is actually been preserved. And importance of this DNA replication is extremely, because if you inhibit DNA replications, in fact that is one of the very efficient way of killing the cells. In fact, you could have designed DNA replicator inhibitors, and you could completely switch off the cells replications and immediately kill the cells. And how fast is the DNA replication is for in humans, for eukaryotes like humans and complex like organisms like us, the rate of the replication is approximately about 40 to 50 nucleotides per second. But if you take a prokaryotic genome or prokaryotic DNA, bacterial DNA, the genome is even more simpler and the replication rate is about 10-fold faster. It's about 500 to 600 nucleotides per second. So just imagine that the rate of the DNA replication is in a way kind of interprets us in a way that tells us the size and the complexity of the DNA. So simpler genomes are actually been replicated faster, but complex genomes like the eukaryotic and the human genomes are actually been replicated much more slower. So the kinetics of the process is actually been way different. Now this is DNA structures, and you could see that when the DNA replications, we know that the DNA structure actually runs in two different directions, the 5' to 3' directions and the 3' to 5' directions. And when you say prime, it means that there are different groups, functional groups which is actually present. 3' hydroxy group is very important because that's the nucleation point from where the synthesis of these incoming nucleotides actually takes place, or the incorporation of the incoming nucleotides actually takes place. If you look at the strands of the DNA, they are actually wrapped together. And once the two strands of the DNA are actually wrapped together, then the next important responsibility of this DNA replication system is to make sure that the two strands actually will be separated from each other. Now, why they are wrapped in the first place? The reason is that once you wrap the DNA, then that's an efficient way of packaging of the DNA. Because if you see the volume of the nucleus, it's very tiny, right? And the DNA molecules are gigantic, very big, large macromolecules. How do you pack such a large molecules into such a small space? One efficient way of doing it is to take the DNA and pack it into smaller bundles, and then put those bundles into those nuclei, and that's how exactly the DNA is actually packaged. Now, histone proteins actually takes a very important role in packaging of the DNAs, and then there are something called the nucleosomes, structures actually formed when the DNA has been packaged, and so forth. But with the packaging of the DNA, the problem of space is actually been solved. That the DNA has to be accommodated in a definite space. But what comes out, problem of this particular process is that how the information of the DNA will be released, because the moment it is actually been condensed in small tiny space, and then the DNA is crumbled into a very small space. Now the information is actually been there, but in a very concentrated form, and it's been very hard to take all the information out. Only one way to take that out is to take the DNA and then melt it. And one way for doing that is to open up the two strands of the DNA, and that's very important that prior to replications, transcriptions, the two strands of the DNA has to separate out from each other and relieve the informations, whatever they have, so that the proteins, molecules can scan their informations and can actually work accordingly. And this is very important, and this is what is known as the melting of the DNA structures. The melting of the DNA structure is a prerequisitions which for DNA replications, DNA transcriptions, and also different aspects of the DNA repair systems. Now, when we see the two strands of the DNA and we understand that their directions are different, one of them runs in parallel directions, another runs in anti-parallel directions. One operates from directions 5' to 3', the other one runs from 3' to 5'. Once you have this directionality issue of the DNA, the replication becomes a little complicated, because now with the two strands of the DNA, in one of the strands of the DNA, when the DNA is efficiently replicated, the other strand of the DNA cannot be efficiently replicated in one go. So they need to be replicated into smaller installments, and that we are going to talk a little later into what we call as the leading strands and the lagging strands. So the whole thing about leading and the lagging strands is where the DNA replication takes place continuously and where it takes place discontinuously into smaller clusters. So we are going to see a detail about that in the subsequent slides. Now, before the DNA replications process we understood completely, we definitely understood and appreciated something called the cell cycles. So if you take a cells and actually allow the cells to grow in a proper nutrient media, then you see that the cell number increases, right? And this is happening because of the reason that there is something called the cell cycle. Now, when you talk about DNA replication, is one of the events which is actually happening within the cell cycle. So cell cycle is actually a composite phenotype of a cell, that how the cell behaves and how one cell is actually producing two daughter cells, or another daughter cell, so how two cells are actually produced. Now the DNA replication is an internal working mechanisms of doing these DNA replications or cell cycles. Now if you see the cell cycle, it's actually divided into different phases of the cell cycle. G1 phase is the phase where the cell is at a rest and actually kinds of prepares the cells for the DNA replication. So many of the components of the cells actually doubled during that time. The cytosolic component, the mitochondrial components of the DNA, and so forth. Now this is a preparative stage where nothing is happening, the cell is squeezing, but at the same time it is actively trying to increase the contents and making it to double, because it is sure that once the cell actually divides, then the thing has to be divided equally, and therefore everything will become half if they are not becoming double in the first place. Now that after the G1 phase, there is the G2S phase. There is an S phase, which is known as the synthesis phase or the synthetic phase. And this is the phase when the DNA actually replication takes place. And one DNA actually produces another daughter strands for in semi-conservative replications. And once the S phase actually takes place, then we have the G1 phase, which is a phase of pause again, where the cell actually pauses after the replication is over and then looks into the DNA and then very carefully scans for any mutations or changes or any kind of an aberrations in the DNA structure. Because if the DNA is been actually replicated, but in replicated in a false way or in a faulty way, then there is a chance that there will be small mutations or small changes in the DNA sequences which will have a far significant and far gigantic devastating effects on the genome as such. And therefore there are mechanisms which actually scans the DNA aberrations, and if there are aberrations, then there is a repair system that actually kicks in, and that repair systems make sure that that particular aberration region is actually being corrected and a new good copy of the DNA is actually represented, and a whole of these events actually takes place into what you call as the cell cycle. Now this is the replication, the steps of the DNA replication. So the two strands of the DNAs are there and then they actually get separated. So one of the strands, this DNA replication takes place continuously and we call them as the leading strands. And there is the other strands of the DNA where the replications cannot take place completely, continuously. Because of this reason that they had a dilemma or they had a problem with the two strands running in the two opposite directions. Now, another very important thing about the DNA replication which I should mention and you should be aware of this is that DNA replication cannot start de novo. In other words, we cannot have a DNA replications just like that with the help of any other assistance. DNA replication does need the assistance of sets of the RNA as in primers, and we call them as the primase reactions. Through the help of these primase reactions, smaller chains of the RNA is actually produced, and these RNA actually binds complementary to that of the DNA sequences, and from that 3' end of one of the RNA, the DNA synthesis actually takes place. So DNA replications or DNA synthesis, these are the similar terms. It's actually been more useful if you have an RNA template or an RNA primer to direct the things. But if there are no RNA primers, DNA replication can never take place, and that's the big problem. That replication is so much dependent on the RNA populations to functions, because this RNA actually produces small RNA primers, which is actually used by the replication machinery to replicate the DNA. Now this is the two modes of the DNA replication, the leading strands and the lagging strands. And remember, both of the two replication actually takes place simultaneously. And the leading strand is very fast because it actually takes place continuously, and the lagging strand actually takes place with the help of small fragments. And these small fragments, which is introduced and known as Okazaki fragment, after it is discovered by Okazaki several years back in the 18th century, where he kind of addressed this issue and said that look, DNA replication takes place continuously one strand, but it takes place discontinuously another strand. And this leading strand is the leading strand, which is the main strand, and then there is another strand which is known as the lagging strand. And the small fragments of the DNA which is actually produced in the lagging strands are known as the Okazaki fragments. Okazaki fragments are the small strands of the DNA which is actually produced because of the action of a lagging strand based replication mechanisms. And once this is actually been done, then there is the cellular machinery comes in, repairs these gaps, fills the gaps with the DNA help of DNA ligases, and a parental copy of the DNA is actually replicated. So there are two strands of the DNA, very important for us to remember. There are the leading strands and there is a lagging strand. That's also very important to understand that and never forget it, and that the two strands actually takes place and the synthesis is different. One of them is continuous synthesis of the leading strand, the other is discontinuous synthesis of the DNA strands, and we call them as a discontinuous steps, and that actually takes part with the help of small fragments which we call as an Okazaki fragment. So this is the structure of the DNA polymerase. The whole central role played by this DNA replication is with the help of an enzyme that is known as DNA polymerase. Now there is no point in trying to give all the credits to the DNA polymerase, because the entire process of the polymerase is very complex and it actually takes place with the participation of large number of different proteins. Now, as we've seen, that RNA template is needed to initiate the process of DNA replications, and this RNA template is actually produced or will synthesized with the help of a primase systems. Now, similarly, when the DNA two strands actually get separated, then there should be some mechanisms which keeps these single strands of the DNA together, and therefore doesn't allow them to reform. And this is what we call as the single strand binding proteins or SSB proteins. The main function of these proteins is to bind to those individual strands of the DNA and stabilizes them and make sure that these two strands doesn't collapse and again reforms the double stranded. because this is very important that the two strands are still separated. And again, when the separation of the two strands takes place, then this actually needs the action of an enzyme that is known as helicases. Helicases actually helps in unwinding of the DNA because the DNA is wounded together, and helicases actually does this function. So you could see the participation of DNA polymerases, helicases. We also see the participation of the primases, and this process actually needs energy. And when the DNA replication takes place, you understand that we just discussed that the small okazaki fragments which is actually being produced after they are actually been synthesized the DNA, there is a small gaps in between these two fragments, and there is a DNA ligase system that actually comes in and fills the gaps, and then actually forms the continuous strands from the okazaki fragments. So therefore, it also needs the participation of DNA ligase kinds of enzymes. So it's a very complex process. In fact, that actually needs the participation of over 100 different proteins in a very orchestrated manner, so that the replication can take place efficiently without any kind of an error. This is again the mechanism that actually shows how the DNA replication is actually taking place, starting from the helicase activity, starting from the unwinding of the DNA, and then the binding of the different primers by the primosome complexes, and then the DNA gets replicated when the DNA polymerase actually sits on that and then actually starts polymerizing it. Another very common thing when DNA replication happens is that there is something called the mismatches. So there is something like an adenine should be there, and on the opposite side there should be a thymidine, because A always spaces with T, but sometimes it may be mismatches, and those mismatches are actually picked back by the DNA repair system. So I just wanted to say that there are different kind of DNA repair systems into play, and all of these repair systems are also very crucial and makes sure that the fidelity of this DNA replication process is well maintained and the integrity of the DNA is not been compromised. So together it needs the participation of large number of proteins and enzymes to manipulate the DNA replication process and to take it and to complete it successfully. Now DNA replication. So the differences between the prokaryotic and the eukaryotic DNA replication. Now when you study the DNA replications, remember majority of the information actually has come from studying the DNA replications of bacteria and also of the yeast. And using that knowledge, actually we understood the DNA replication mechanisms in humans. So there are something which is very common for these two classes of organisms, the prokaryotes and the eukaryotes. A few of the features are there which are very unique for the prokaryotes, doesn't express in the eukaryotes, and vice versa. So we need to understand that differences, because that difference is going to be very helpful for us. Remember that DNA strands, we are very sure that we have been aware of this basic fact that the DNA has 4 different nucleotides, right? Adenine, guanine, cytosines, and thymidines. So A, T, G, and C. And there is a distinct pattern of base pairing between these bases. So A always pairs with T, C always pairs with G, and A and T pairing is carbon double bonds, and there is one sigma bonds and there is one pi bonds. But the kind of bondage between cytosine and guanosine is G and C triple bonds. So there is one sigma bonds and there is 2 pi bonds. So the A and T bonds are a little easier to break as compared to G and C bonds because they are triple bonds, right? Now, when the DNA replication takes place, then there is a local melting of the DNA. We use the word known as melting, where the 2 strands of the DNA gets melted by the help of an energy-driven process, and also driven by the helicases and other kind of driven DNA assisting proteins or replication assisting proteins. And when the 2 strands actually get separated or melted, then this ease of melting is more easier to the places where there is more of A and T as compared to G and C. Just for the reason which I mentioned just now, that A and T has double bonds and G and C has triple bonds. So triple bonds are difficult to break, A and T are easier to break. So the melting temperature or the melting energy is much less than for an A and T melting as compared to G and C melting. So majority of the places where DNA replication initiates are reaching A and T. But just because there is an A and T doesn't mean that that particular region will be an epicenter for DNA replication. So just be aware of that. Another very important thing about DNA replications in prokaryotes and eukaryotes is that the eukaryotic genome is rather smaller, and therefore there is one replication that initiates, we call them as a replication bubble, and that transmits just like a wave and spreads to the entire plasmids or the entire DNA of the prokaryotes. So just one replication bubble is enough to initiate the replication in a prokaryotic system. But for a eukaryotic system with billion base pairs, it's not easy for one replication bubble to initiate the process of replication, and therefore multiple replication processes actually initiates, and we call them as firing of the replication bubbles. So replication bubble is the point where there is a local melting of the DNA, and the DNA gets separated into 2 different strands. And then this process doesn't initiate at one place in the case of eukaryotes, but actually takes place and originates at multiple places, and we call them as firing of replication bubbles, because this actually looks like a bubble because it actually swells up and the two strands actually get separated. So this is another very important thing, is that there are multiple replication origins in eukaryotes as compared to only one in the case of prokaryotes. Now prokaryotes and eukaryotes also differs in the DNA polymerases which they have. When the prokaryotes are there, we call them as in polymerases 1, 2, 3. If the prokaryote, if they are eukaryotes, they have the different polymerase sets, the complexity is different and so forth. So the DNA replication initiation is also very important, that how the process is initiated. I just discussed below and a few minutes back that this DNA replication initiates with the process of melting of the DNA. And once the DNA is melted, the two strands are separated. There is an SSB protein that goes and binds to those strands, stabilizes them, and initiates the process of daughter DNA synthesized, or the second strand of the DNA being synthesized, using that as a template strand. Now after the DNA has been initiated, then there is an addition of the nucleotide triphosphates in the form of DNTPs to those 3' hydroxyl groups of these primers, or of the nucleotide, existing nucleotides, and then the DNA actually stands actually elongated. The elongation means the copying of the whole DNA end to end, so that the entire region gets copied. Remember always that the ends of the chromosomes, that is known as telomeres, are something very hard to copy, and there are different mechanisms to copy those regions, because if you don't copy it, none of the cells will be viable, because with every generation the telomere length actually reduces. So unless or until there is a mechanism to replenish that particular length with the DNA replication of the telomeres, then it's not going to work, and the cell is quickly going to the state of senescence. And that exactly happens in the case of cancerous cells, is that they are immortal because there is a telomerase activity. And because of that telomerase activity, the DNA strands or the DNA chromosomes remains intact. It doesn't get reduced from generation to generation, and the cells are actually viable and can actually replicate the cells. The regulation of the prokaryotic replications. So the regulation of the prokaryotic replications, there are different mechanisms for that, and we are not going to detail into too much because this is a very standard understanding in the textbook. So just feel free to go and read about it and then find out that what are the regulations of the prokaryotic replications. But pretty much the mechanisms that actually regulates prokaryotic replications is kind of similar, if not identical, to the eukaryotic replication systems, and that is very helpful for us to understand that there is a universal common rule which actually operates in order to replicate the DNA across all the different classes of organisms, whether it is prokaryotes or whether it is a eukaryotic system. Now, once the linear DNA is actually been formed, because in the case of eukaryotic systems, the DNA is actually been coiled, it has to be unwinded, and the linear DNA is actually produced. And once the linear DNA is actually produced, then it actually goes to the different stages. Right? There's the pre-initiations, then there is an initiations, and then there is an elongations. And after elongation, there is something called the termination, which is very important. Another very important thing about DNA replications is that, remember that DNA replicates only once per cell cycle. So if there is a replication bubble which is initiated in the eukaryotic system, then no other replication bubble should be there in the systems in the next phase of time. So at any given point of time, it is just one replication bubble that actually initiates, and that actually initiates the process of DNA replications. Now this happens in prokaryotes, but in eukaryotic genome is so complex and so large that you just cannot replicate the whole genome, and therefore multiple replication bubbles are actually produced, and multiple replication firing actually takes place. Another very important aspect of the DNA replication is that per cell cycle the DNA is replicated once, but just once, not even twice. But whereas we should remember that there's a universal rule that DNA has always been replicated once and only once, and this is to ensure that we don't have additional ploidies and copies of the DNA sitting into the cell. So this is a cartoon that actually shows the Sing virus 40 replication mechanisms, its tools and techniques. So again, it's the very self-explanatory slide. So feel free to go and read the detail about the slides, and you will be actually amazed to see that and how the intricate mechanism of the replication is actually worked out with finite details. The regulation of the eukaryotic replication. The regulation, as I mentioned, is a complex process because it actually involves the participation of the different proteins. And all of these proteins actually plays a very significant, even though it's a small role, but very significant in trying to moderate the entire replication process. Now, how to know that the DNA is actually replicated? So there are different tools and techniques and wet lab experiments to confirm that the DNA is replicating or not, or whether it's been replication competent or not. And one way of doing that is that labeling the DNA with the help of different dyes, like for instance Ki67 index, which people always do, or very commonly do for cell cycle analysis, is that when they stain this dye, the cells becomes little colored, and once they looks little colored, we can take to and flow cytometry and going to analyze the DNA. Now remember this kind of an analysis of the DNA, and there is other methods like Ki67 is going to give you an index of the DNA replications. There is something called the BRD incorporation, so thymidine incorporations, where when the DNA replicates, Then instead of the nucleotides, we are giving them the radionucleotides, and once the radionucleotides are formed, now you can quickly image the systems or can monitor the systems. So that is another very efficient way of studying this particular process of DNA replications. Now the fidelity of the replicator, this is very important, because what happens is that the replicators for the DNA replications should maintain that the replication takes place in a non-faulty way, and if there isn't faulty way, or if the DNA replication produces certain kinds of mismatches, then there are different systems of the enzymes that actually sits in close proximity to the DNA, and about 300 to 400 in number, and they monitor the systems very carefully, and if there is a system aberrations, then they quickly informs that the DNA replication is not correct, or it's faulty. So this kind of mechanisms with help of proteins is an ideal example where you see that protein DNA interaction takes place a very crucial role in moderating the entire process of the DNA replication. So coming to our summary, the DNA replication is a complex process. It actually involves copying of a single strand of DNA into the daughter cells. We also found out that how the DNA mechanism of the DNA replication is actually worked out, but people were a little clueless before they actually discovered, or someone has discovered, that the DNA can act as a genetic material. And we have seen how with the help of delegate experiments this is actually been addressed. Also, that is very important for the DNA replication is to know the structure of the DNA. We know that Watson and Crick actually solved the structure of the DNA using an X-ray crystallographic data, and from their information we come to know that the DNA is a double-stranded molecule. And once it is a double-stranded, we also further know that the two strands actually complementary to each other. So in other words, one strand of the DNA has enough information to predict the structure and the functions and the sequence of the other strands of the DNA. And once this particular mechanism is operational, we know that the genetic material is truly the DNA. Also, we understood and we have read in this particular class is that there is a semi-conservative mode of the DNA replications where the two strands of the DNA doesn't completely produces two new strands. Rather, it is always the parental strands hybrid with the new strands and forms an hybrid and a daughter strand. And this is very important for us to know that this is proved beyond a reasonable doubt with the help of solid experiments that DNA replication does take place in a semi-conservative mode. Also, we have actually studied that how the replication actually initiates in a process or in a place that is known as the replication bubbles, and those are actually the bubbles which are fired during the process of replications. In eukaryote, you just need only one bed, or in prokaryotes, you just need only one bubble, but in eukaryotic systems as complex like humans, multiple replication bubbles are actually initiated, and the DNA is actually gets replicated from these bubbles. Remember, we also discussed about that the DNA has nucleotide sequences with A pairing with T, G pairing with C, and majority of the time, or of all the time, A and T base pairing is double-stranded and C and G base pairing is triple-stranded, and double strands breaks are much more easier to break as compared to that of the triple strand. So majority of the origin of the DNA replications are AT reach. While we say that, we also want to put in caution that just because a sequence of the DNA is AT reach doesn't mean that that is going to be a hub of the DNA replication. It is much more complex, and we have actually not understood completely of that particular scenario. Also, from the DNA replications, we understood that how participation of the different DNA assisting proteins are crucial in modulating the process of the DNA replications, because it's just not the RNA or the DNA polymerases, but assisting of the different proteins like topoisomerases, like helicases, like the different kinds of the proofreading activity, enzymes with the proofreading activity related to the DNA repairs, and the DNA ligases, all of them together actually orchestrates the process of the DNA replication. Because there is a fallout of this, if the DNA is not being replicated properly, then it can quickly activate the different cellular pathways, or the DNA may, or the cells may undergo actually apoptosis or program cell death. So the DNA replication is crucial. Understanding the mechanism of this is actually fundamental in understanding molecular biology with an even greater depth and precision. Thank you so much.